Then they talk about the principle of water moving from one place to another, high to low concentration still. What's that called? Osmosis. And then we talk about the principles of tonicity. Now, thinking of tonicity, I'm thinking of water movement from one place to another. If I have too many dissolved substances outside the cell, dissolved in water, I have what we call a hypertonic solution. Now, where is water going to want to go? It's going to rush outside the cell to equal out the concentration of water on either side of the cell. What's going to happen to the cells? They're going to shrink up. What if I have a hypotonic solution? A solution that doesn't have as many dissolved substances as what I find inside the cell. What's going to happen to my cell? Yeah, it's going to blow up because water is going to move from a high concentration outside the cell into the cell. What's going to happen to the cell? It's going to fill up with water and burst. So what's the perfect scenario with respect to water movement in and out of cells? Isotonic solution. And an isotonic solution has the same number of dissolved substances inside and outside the cell. So water is going to move in and out at an equal rate. And that's going to be happy cells. Yes? So understand, page 71, the principle of tonicity. Now, some things have to move across the membrane and need help and energy to do so. And what do we call these? Yeah, this is active transport. So I need actual molecules that are hanging out in the plasma membrane to physically grab onto something and bring it in, or physically grab onto something and push it out. That's an active process. That's going to take energy in the form of what? What's our energy molecule? ATP, adenosine triphosphate. So they describe a couple of different scenarios with vesicular transport, actually creating a little package around something and pushing it out, or creating a package around something and bringing it in. What are the two terms we need to know for that? Endocytosis and exocytosis. Ooh, we're getting blueness. So endocytosis would be what? Grabbing something and bringing it in. Exocytosis, put it in a package and put it out. So that's vesicular transport. We need to know that. What's phagocytosis? Phagocytosis. Yeah, I'm going to reach my little plasma membrane arms around something, wrap it around that something, and do what? Bring it in. Now, what if it's a bacteria? Yeah, I'm going to bring it in and do something to it. But that's what phagocytosis is. I can also put my little cell membrane arms around things and bring in extra fluid. Pinocytosis, that's the other term. Again, these are active processes. That means they what? Need ATP to happen. Very good. Sometimes I have little receptors on the surface of the cells. And when they come in contact with things, it's going to cause these little packages to be created. What do we call that? Receptor-mediated. So receptor-mediated endocytosis. There's some little receptor come up on the screen. I'm going to kiss you. No, I'm not going to kiss you. So, something comes in contact with my receptors, it's going to cause me to create that package and bring it into the cell. You can see this on page 76, figure 3.12. On page 78 in your textbook, table 3.2, kind of gives you a summary of all these things, okay? These are all active membrane transport processes. These all take energy to happen. We're going to talk about things like the sodium-potassium pump. What is that? Why does that take energy to happen? Well, I'm pushing things against it. I'm working against the concentration gradient. I'm actually taking sodium and pushing it where there's higher concentration of sodium. That's going to take energy to happen as well. So when I work against what should normally happen, and we'll see that in our cells, that's going to take energy to happen. So those little sodium-potassium pumps are an example of active transports. Plasma membrane and membrane potential. When I talk about the plasma membrane, it's got a charge associated with it. It's negative on the inside and positive on the outside. One of the reasons is because of that sodium-potassium pump. Okay? So it talks about why we have that membrane potential. When we talk about the nervous system, we're going to talk about how important that charge is. And when that charge flips, it's going to cause the cell to do something different. So that membrane potential is important. The membrane at rest is said to be polar. What's that mean? We talked about a polar molecule, right? No. It's what? It's got two opposite charges, right? Positive on the outside and negative on the inside. No. Don't think middle. Positive on the outside, negative on the inside.
Well, I'm just thinking of like how the membrane would be with a polar heads on the outside because of the negative charge.
Yeah, but that's a different concept. It's water loving versus hydrophobic, and that those heads are called polar heads. Yeah. Correct. The phosphate end of the molecule. Yeah. Okay. Good. They talk about adhesion molecules, molecules that hold membranes together, CAMs, cell adhesion molecule. Now what does it say? Yeah, but it's not that, it's this. It's not my computer. Kick it. That's what's happening. It's not communicating. Heck with it. You'll get it in the video. I know I switched back and forth. I plugged it in. I don't know what else to do. What else do I do? This is working, yeah. What do you mean, unplug it from here? Screw it. Unscrew it. I'm sorry. It's going to be on your video, and I'll refer to the pages so you can see. Okay? Just easier, and I'll yell at IT later. This room doesn't seem to ever work. Okay, cell adhesion molecules. These molecules that we talked about when we talked about plasma membrane not only serve to do all of those jobs that we discussed, but they can also be street signs for the cells as well. And that's what they talk about under these roles of membrane receptors. So they can be little bridges, or they can be pumps that pump things in and out. But they can also act as chemical signals from one cell to the next, and what we call contact signals. And they can be basically, in layman's terms, street signs. This is who I am for other cells. When we talk about the immune system second semester, we'll talk about how does a cell recognize that a cell belongs to me. And it does that by using these cell recognition molecules. Yeah, carbohydrate groups that are attached to these proteins. We call them glycoproteins. Okay? Cytoplasm. What's inside the cytoplasm? That's where the book goes and talks about all the different cell parts. So you should know a basic definition of all the organelles. And what's an organelle? Yeah, give me the definition for little organs. Little parts of the cell that have different jobs to do. It does. S phase. I'll talk to you after class. You interrupted me. I'm going to beat you like a stepchild. Look under cell cycle. It goes through all the phases. And then look where it defines cytokinesis. So, the book goes on and on and on about each of the organelles. Now, each of the organelles has a job to do. So when we think of the cell, think of it as a little factory. And every little part has a different job to do to run that factory. I'm trying to find you the nice, oh, here it is. On page 94 and 95 in your textbook, they do a very nice job of doing what I call making information small. So there's all the organelles, and it defines what they all do. So that's a better place to look, right? Instead of reading 26 pages about all the organelles, I got 2. Better. So mitochondria, ATP, powerhouse of the cell. Ribosomes, that's where I'm going to put together my proteins. It's the site of protein synthesis. The rough endoplasmic reticulum. Endoplasmic reticulum's like streets within the cell. I'm going to transport things from one place to another. Smooth endoplasmic reticulum, same thing. Golgi apparatus. That's where I'm going to package things up and ship them. I call it the UPS system of the cell. Packaging and shipping. Remember we talked about exocytosis? Okay, well, how do I get stuff out of the cell? I put it in a what? I put it in a package. That package is phospholipid. It's the same as what my plasma membrane is. And then I use this little analogy. Those of you who have already had me, bear with me. You'll hear it again. But when I have to get inside and outside a submarine, what do? Does anybody know? I'm not just going to go open the door to the submarine while I'm underwater and say, okay, I'm leaving now. Because what's going to happen? All the water from outside is going to rush in. So how do I get out of the submarine without drowning everybody in the submarine? Yeah, I go into an airlock, exactly. So I have this special little compartment that I go into, and I shut the door behind me, and then I open the compartment, and what happens? Water rushes in there, and I can leave. So remember exocytosis? Right. I'm making a package. Who's going to help me make the packages? The Golgi apparatus. So things can leave the cell without blowing a hole in the side of the cell. Peroxisomes. Packages of chemicals. I put them in packages so they don't destroy the rest of the cell. Remember that bacteria I phagocytized a little while ago? How do I kill it? I merge the package I put the bacteria in with the package of chemicals. They come together, and what's going to happen to the bacteria? Boom. But it's in a package, so it doesn't destroy the rest of the cell. Then what am I going to do with that package? Exocytosis, throw the trash out. Who's going to take care of all the dead little bacteria bits?
Does it go through the Golgi again?
No,
Before it goes out?
not in this case. What am I going to do with the dead bacteria bits I threw out? Yeah, the circulatory system will bring them to the places to get rid of the trash. Lysosomes, same kind of thing, different chemicals. So basically I can create a vacuole, a package, and I'm going to call it something different depending on what's inside. So if there's peroxide-like chemicals in the package or vacuole, I'm going to call it a peroxisome. If there's other chemicals that are going to blow things up or lyse things, what do I call the package? A lysosome. Then I have a whole bunch of different proteins in different configurations that are going to help to give my cell shape. And that's what we are described as microtubules, microfilaments, intermediate filaments, and then centrioles or organelles that actually make these proteins. These are all part of your cytoskeleton, your cell's skeleton. And their cell skeleton are basically these protein structures. Then we can have little extensions of the cell. We can take these little microtubules and make these cute little extensions called cilia. We have some big long ones that will actually help the cell move from one place to another called flagella. And then little extensions on the cells themselves called microvilli. All of these are different configurations of these proteins, and they're going to help with movement, either the cell from one place to another or things across the top of the cell. What's the nucleus? Control center. Everybody says control center. What's in the nucleus? DNA. Deoxyribonucleic acid. And what's that going to do? It's going to be the blueprint for all the proteins that that cell's going to make. That's your control center. It's going to tell the cell what to do. The nucleolus makes molecules that are going to help that DNA do what it needs to do, called RNA. Okay? DNA and RNA are both what? What kind of organic molecule are they? They're called nucleic acids, and they're made of nucleotides, right? Those nucleic acids are the basis of protein synthesis. The next part of the book talks about cell growth and reproduction. And this talks about the different phases of a cell's life. This is where you're going to find your information. So, you were born, yes? When you were born, what did you have to do? Grow up, right? And then we go to school, we get a job, we do what we're supposed to do for a living, right? And then someday we say, let's make more. Yes? Okay, think of that when we think of a cell's life. Because a cell is born, and then they're going to live their life. That live their life part of a cell's cycle is what we call interphase. So during interphase, after I've been born as a cell, I'm going to hang out and grow, and I don't know, if I'm living in the pancreas, maybe I'll make some insulin, maybe I'll make some glucagon, maybe I'll do some stuff for a living. And then at some point, we decide to make more. We want to reproduce that cell. In order to reproduce the cell, I have to make sure that all the directions I have to do what I'm supposed to do for a living go with the new cells. And what were the directions? Where were those located? In the DNA. So, during the S phase of interphase, I'm going to make a copy of all my genetic material. Now that I have 2 copies, I'm ready to make 2 cells. Then we go into, after the G2 phase, and this is where I kind of proofread all the copies I made, make sure they're good. I go into what I call the mitotic stages of my life. And this is where I'm going to take all that genetic material and evenly separate it to make 2 brand new cells. So again, I'm reading on page 96, 97. So what are the different stages of mitosis? Prophase, metaphase, anaphase, telophase. Near the end of those mitotic stages, not only do I have to separate and copy genetic material, but I also have to make sure my brand new cells have what? Because genetic material isn't the only thing in my cells, right? What else is inside my cells? Mitochondria, ribosomes, Golgi, all those other things. So near the end of this split, end of anaphase and through telophase, I'm going to replicate my organelles. It's on page 97. Now, when I replicate the organelles during the end of anaphase and telophase, I'm going to continue that after the cells have actually separated. That is called cytokinesis. So if you look at the diagram on page 97, figure 3.31, you can see cytokinesis in the yellow, but it kind of has a little tail that goes in through anaphase and through telophase. Okay? So if I ask you to define what cytokinesis is, it's the replication of the cellular organelles. Okay? So know what happens during S phase, G1, S, and G2 of interphase. Understand that it's important to replicate the genetic material so that both of my brand new cells that are created after the mitotic stage have the same directions. On page 98, we see a picture of the replication of DNA. And basically what I'm going to do is take that double-stranded group of nucleotides, split them, and make a copy of both sides. If I make a copy of both sides, remember, only certain nitrogenous bases can come together, right? Remember we talked about that in the chemistry chapter? Adenine can only hook up with thymine, and guanine can only hook up with cytosine, A, T, C, G. And those free little nucleotides that are going to help make the new strands of DNA are floating around in the fluid of the nucleus. Okay? Those enzymes, and those enzymes are like those big blobs that you see, are called polymerase enzymes. And basically what a polymerase enzyme is going to do is to bring things close enough so they can do what? Come together. So a DNA polymerase enzyme will basically bring those free nucleotides in close enough to the strand that already exists so they can do what? They can adhere and hook up. That enzyme is going to be like a little knitting machine that's going to move its way down the strands, and it's going to help to create 2 identical strands of genetic material from that one. Okay? So DNA replication takes place during the S phase of interphase, and then when I move from my house and I move into a new apartment, or go from a new place to another, what do you do with all your stuff? No, think about what you do. I'm ready to move. What am I going to do with my stuff? I'm going to, because it would be a pain in the neck if I just took everything and just kind of threw it into the car, right? So I'm going to take it and I'm going to package it up. The first phase of mitosis, I'm going to take all that genetic material. Now I have a whole bunch of strands. How much genetic material does the normal cell have? How many chromosomes, do you know? 46 chromosomes. By the end of S phase, how many chromosomes do I have? 92. Woo! They're all over the place. So I'm going to package them up during prophase. And this is when I actually start to see chromosomes. You can see them under the microscope. You guys probably looked at them in lab already. Have you looked at them in lab already? Okay. When you look at the mitosis of whitefish, you can see these great pictures. You won't see these under the microscope. These are fancy schmancy stains and fancy microscopes. But on page 100 and 101, you can actually see the phases. Okay? So during prophase, I'm going to pack my stuff. I'm going to package the chromosomes, pull them all together, make them tight, and hook them up so that one copy's on one side and one copy's on the other side. And I'm going to glue them there with some little proteins. During the next stage, and in your book they show us early and late prophase. Prophase is prophase. Early prophase, we still have a plasma membrane around the nucleus. What happens by late prophase? The membrane's gone because it's just basically going to get in the way. So, packaging chromosomes. Then I'm going to take them and I'm going to line them all up in the middle. Metaphase. And you can physically see them. It kind of looks like little spider legs, like this. And they line up in the middle. Those centrioles that made those proteins for me, they're going to make these little sticky proteins that are going to help pull the two copies to opposite sides. Huh? Yeah, well, the metaphase plate is them lined up in the middle. See all these cute little green things that are running down? They're called spindle fibers. The centrioles made those for me. And I equate them to fishing poles. So when I'm going fishing, right, I get my fishing pole out with my fishing wire on it. And I put a little bait on the bottom. And I throw it into the water. Spindle throws itself out and grabs onto one side of either side of the chromosomes, right? And what happens when I catch a fish? What do? I reel it in. I shorten the string. Guess what happens to the spindle during anaphase? It's going to be reeled back into the centriole that threw it out. And it's going to pull anaphase away. It's going to pull those pairs to opposite poles of the cell. You still with me? So anaphase, we're going to pull them away. Now I'm going to start to replicate those organelles and get ready to make two brand new cells. That's going to happen during what? Telophase. So cytokinesis, replication of the organelles, that's going to start happening in anaphase. During telophase, I'm actually going to pinch those plasma membranes around the two brand new groups of genetic material that I have now that could pull during anaphase. And I'm going to create two brand new cells. What do you think is going to happen once telophase happens? Well, what do you do when you move into your new apartment? I'm going to unpack. So I'm going to unwind that genetic material. I'm going to create a brand new what? Nuclear membrane around that genetic material. I'm going to do my growing thing, and then I'm going to be a brand new cell with all the directions I got from my original cell. What do we call me now? They're daughters. We call them daughter cells. So those two brand new cells, well, once they unpack and get everything going, just like you in your new apartment, right? They'll get down to business once they get their organelles replicated and all that good stuff. Okay? So understand life cycle, understand metaphase, anaphase, all those good things. Okay. The last part of the chapter talks about the role of genetic material. And the role of genetic material is basically to make proteins. I make my proteins based on my genetic code. And my genetic code is what? Yeah, but what about my DNA? You have DNA, you have DNA, you have DNA. Do we have the same code?
No, it's unique to each person.
It's unique to each person because? But why are you unique? not the amino acids. I got no marker. Damn it. What's different from my DNA than your DNA?
Just the way those letters are arranged.
What letters?
Yeah.
Nucleotides. Very good. So the order of your nucleotides. Now you're human, you're human, you're human, you're human, right? Most of the time, except on Fridays and Mondays, I'm kind of coming back. What makes me different than you? Most of our genetic material is the same. Did you know that? It's about 1.5%, 2% that's different. It's the order of nitrogenous bases. My A's and my T's and my C's and my G's. That's what makes me different from you. Yes? We're all still human, so most of our A's and T's and C's and G's are in the same order. So what are some of the things that are different about us? Eye color, hair color, size of our body, skin tone. That's the stuff I'm talking about. Those proteins are made from your genetic code. And your genetic code is the order of nitrogenous bases in your DNA. Now, I think probably you got this a little bit in one of your videos. But think of the DNA as the big boss. Yes? And you did get that, didn't you? Okay. Now the big boss is too big to leave his office. But he's got to give everybody orders, right? DNA is too large a molecule. It doesn't leave the nucleus. But I have to get directions from DNA in order to make my proteins. So who's going to go into the big boss's office? The secretary. RNA. And the RNA is basically going to get the code. RNA has nucleotides, just like DNA did, right? Except what's different about RNA? Yeah, we have a different nitrogenous base, uracil, and it has a different what? Different sugar. But those little nucleotides, little RNA nucleotides, are floating around the nucleus too. When I want to make a copy of the information from DNA, I have to unzip it. So if you look on page 103, we see enzymes that are going to help unzip the genetic material and read the code. When I say read the code, what am I reading? The order of nitrogenous bases. So I'm going to make a copy with RNA, messenger RNA, when I'm finished, that's going to take that information and bring it outside the nucleus so I can make my proteins. So with the help of polymerase enzymes, except now I'm making an RNA strand, what do we call the enzyme? It's called RNA polymerase enzyme. Remember when I was making DNA in S-phase? What did I call the enzyme? DNA polymerase. When I'm making RNA during this process of copying, by the way, it's a two-step process. It's called transcription and translation. Yes? So when I'm in transcription, when I'm copying, I'm the secretary in the boss's office. I'm going to create a copy of what's on the genetic material, a mirror image of it in RNA. Once I'm finished copying the information, it's only a small portion of the DNA, by the way. I'm going to then take that strand, that copy, the secretary is going to leave her office and go to her desk and do what? Translate. The messenger RNA that I've created a copy of the information from DNA is going to leave the nucleus. And where's it going to go? It's going to go to out the endoplasmic reticulum and hang on a ribosome. And the ribosome is like a little knitting machine. It's going to hold the messenger RNA in place, and with the help of another kind of RNA, which is called what? tRNA or transfer RNA, I'm going to knit myself a protein. If you look on page, I don't know what that page, 106 and 107, we see this whole thing in an image. For every 3 nitrogenous bases, when I'm making a protein, that's going to equal 1 amino acid. Now the copy I made from DNA with messenger RNA is a series of nitrogenous bases. For every 3 nitrogenous bases on that messenger RNA, and what do I call those 3 nitrogenous bases? They're called codons. I'm going to have a complementary match floating around transfer RNA, which also happens to be carrying an amino acid. The 3 complementary nitrogenous bases on transfer RNA are called anticodons, and they are only going to hook up A's to U's or C's to G's in a very specific pattern. If you look on page 104, we're going to see figure 3.36, and we call this the genetic code. Well, what is this a map of? Well, no, there's a start and a stop to every single protein, so there's one of those codons that are going to give us information about starting my protein and stopping my protein. What is this a chart of? Codons. So if I take that messenger RNA and I have UUU, what transfer RNA is going to come and hook up with it? AAA. Now this is a chart of codons. So which amino acid do you think is going to be stuck in that spot? You're going to find UUU, because this is codons, not anticodon. What is it? Phenylalanine, PHE. There's 20 different amino acids. Do you see this chart? This is going to determine which one goes in which place. So the codon is on the messenger RNA. The anticodon is on the transfer RNA. The transfer RNA is also carrying a specific amino acid. And that's what's confusing about this chart. This is a chart of codons. So this is the pattern that I'm going to see on the messenger RNA. I wish they would put the anticodon. That would kind of make more sense if I was looking at it. But this is what I find on the messenger RNA. Its complement will hook up to it. There's 3 little places on your ribosome. So once I make a messenger RNA, that messenger RNA strand is going to go out to the ribosomes. The ribosomes are usually sitting on the endoplasmic reticulum. What do we call endoplasmic reticulum with ribosomes on it? Rough ER. And now I'm going to start the knitting process. So with the help of the ribosomes that kind of act like placeholders and push things through, I am going to undergo protein synthesis. The transfer RNA is going to bring in those specific amino acids into this little knitting machine so that they can get close enough together to do what? Connect. There's 3 little places, the E site, the P site, and the A site. So the messenger RNA, and again, if you look at that picture on page 106 and 107, you can see it Huge and big on page 106. So the messenger RNA is basically going to lock itself into a ribosome. And the ribosome, which is also made of RNA, is going to help move the strand along. It's going to enter and it's going to start the process. In the P site, that's the place where a transfer RNA is going to come and dock. Now look at the code in your book. The anticodon is on the transfer RNA, that thing that kind of looks like a little body, and then there's a little thing sticking up with a little circle on it. Everybody with me? Okay, so this is the transfer RNA. It's only going to lock into its complementary codon on the messenger RNA. So the transfer RNA's code is UAC. Everybody see that? What's the messenger RNA code? AUG. That's the only one that can fit there. Yes? And if you go look up AUG on your chart here, which of the amino acids are you going to see? Methionine. Yes? Making sense? Who's totally lost? Who will not admit they're totally lost? So that's what all the code, that's what that whole code thing is. It's telling us where they're going to fit in the scheme of things. In the A site, another one of those transfer RNAs is going to come in. And when they come in right beside each other, guess what's going to happen to the two amino acids hanging at the top? Then now they're going to get close enough to do what? Link. Through what kind of chemical reaction? It's going to make a polypeptide, but what kind of chemical reaction brings the two together? Synthesis reaction. What kind of synthesis reaction? Yeah! Dehydration synthesis reaction. Suck out some water, stick the two together. Yes? Now the whole strand is going to pick up and move over one. Now the original P site is going to be in the E site. I call it the exit site. We're going to kick off that transfer RNA, but what are we going to leave behind? The amino acid that was hanging at the top, because it's stuck to the other one that is now in the what? It's in the P site, yes? Another transfer RNA is going to come into the A site. Guess what's going to happen? That's going to stick to the other amino acid, the one that was in the P site. The whole strand's going to pick up, move over one. We're going to kick off the transfer RNA. Yes? We're going to leave behind the what? Amino acid that was attached to it. You getting the point? That's going to keep happening systematically. That's why I call it a knitting machine until we get to the end. When we get to the end, we've created a strand of what? Amino acids. What are we going to call it? A polypeptide, yes? And it's in a very specific order. Based on what? Your DNA. Yes? The RNA helped, but it read the code. Do you see where we're going? So transcription and translation, and I'm a very visual person, so I love this particular diagram because I can see all the steps, is going to involve unzipping the DNA, making a copy. Secretary goes into the office, makes a copy of the information from the boss. We make a strand of what? Messenger RNA. That's the copy. Then what am I going to do? Messenger RNA has to detach and leave the nucleus and go park itself on a what? Ribosome. Then, based on the codon, we have transfer RNA that's going to hook up with it with the help of an enzyme and leave behind its amino acid. That's called translation. Okay? Yes? So the book gets a little crazy with the whole process, and I wish I could show you a video, which would probably be quite helpful. But this process happens with several ribosomes, because when I actually read genetic information from DNA about making a protein, it's quite long. It's many, many nitrogenous bases long. So when I actually go out to translate it in the ribosome's endoplasmic reticulum place, several different ribosomes are going to help me do that. So basically I'm going to read ends, middles, and then the end process, which is going to happen in the endoplasmic reticulum, is going to tack all the pieces together to create the amino acids in order. So the processing of proteins is going to happen in the endoplasmic reticulum. When I'm done making that protein based on the genetic information, I'm going to do what with it? Package it up and send it either to the cell to do some work, or I might send it to the Golgi apparatus to leave. When you had breakfast this morning or lunch this afternoon, hopefully you took in some carbohydrates, right? In order for me to take those carbohydrates into my cells, I need help. I need something called insulin. Where does insulin get produced?
Pancreas.
In the pancreas. But I need it all over my entire body, right? When I get the message that I need to make insulin, guess what's going to happen to the cells in the pancreas? They're going to unzip their genetic material, and only the genetic material where the information is about making insulin. Not all the genetic material, just that part. And what's going to happen? Transcription, translation. I'm going to make a chain of amino acids because insulin is a protein. Yes? Nope, it's a protein. That protein is going to help me do what? Not make sugar, use sugar. Okay? So that cell, going to transcription and translation like crazy, make a whole bunch of insulin, package it up, send it to the Golgi apparatus. What's the Golgi apparatus going to do? Package in, ship it out. It's going to go into the circulatory system all over the body and help all my cells do what? Use. Because of receptor-mediated endocytosis. It's either, there's several different types of diabetes, so I might have a problem actually making insulin. That's a problem. Or it might be the receptors on my cells that are misshapen, and they don't know how to use the insulin, because it's kind of like a lock and key thing. So that's another reason why. Did you have a question, hon? In the human body, billions. How many different types of amino acids are there? 20. So, you ever play the lottery? Nobody plays the lottery? Come on. I play Powerball every week. Are you kidding me? How many different numbers on the Powerball? Well, no. How many different can you choose from? No, it's like 40 numbers, right? Yeah, so how come you guys don't win Powerball every week? There's only 40 numbers.
Because of the odds.
Because of the combinations, right? So even though there's 20 amino acids, remember, it's the combination of them that makes the proteins different. And where do I get the information about the combination? Your genetic material. So even though there's only 20 amino acids, did that help answer your question? No, the question was amino acids. So there's 20 different amino acids, and I put them in different combinations, and I can make billions of different proteins. Because my proteins can be of different size. They can be shorter. They can be longer. What were the different protein configurations I could have? Chemistry chapter. Proteins come in what shapes? I'll give you a hint. Primary, secondary, tertiary, yes? So all those proteins in different lengths and different orders of amino acid. The order of amino acid is going to determine how I what? Fold my protein. And if I fold it a different way, I'm going to have a different active site. Different active site, different function. Now, does that make more sense? So I can have billions of different proteins, different combinations. Yes? So, all of the stuff that we see on page 108 and 109 is basically reiterating and showing us the different combinations that can occur based on the genetic information. Have I sufficiently blown your mind yet?
Yes.
Okay. So, questions? Oh, I can hear that. I hear that. You're not saying it. Your eyes are saying it, though. So your test is Wednesday. Your test is on chapters 1, 2, and 3. What's going to be on your test? Everything we've talked about in chapters 1, 2, and 3. And some people say, will you give me a study guide? Study everything we talked about in chapters 1, 2, and 3. There's your study guide. Yes? Now, how can I make myself not scream inside? You should already have done this, but now, if you haven't done this already, do it right now. What's going to help you study the information?
The outline.
Yeah. Go to your chapter outlines that I put up online for you, okay? So that's basically all the stuff we talked about in class. Yes? The important points from the chapters. Understand, if I say no transcription and translation, understand those points, okay? The terminology, don't worry about memorizing the diagrams. That's not me, that's Erica. Yes? So that you're going to find in Erica's class when you have to label the diagram. That's not here. But if I say cranial is in the head, chest, legs, what are you going to say? Head. Yes? So that's the kind of question you'll see from me with respect to those terms. Okay? Chapter 2, chemistry. Right? I can tell. I heard louder screams when I said the C word just now. Remember, do not panic. It's extremely basic chemistry. Yes? I know some of you don't think so, but it is. Also, it's a multiple choice test. Yes? Remember that. So I'm not going to make you reinvent the wheel and create chemical compounds and components. I'm going to ask you questions like, protons have a what kind of charge?
Positive.
You know, that's the kind of question you're going to get from me. Don't panic. 100. Chapter 3, we're talking about cells. So we're going to talk about different components of cells, right? Don't go away. I have more time. Don't tell me I didn't. Oh, my God. Like, so I bet you I put it on my desk. Son of a... I got the box. I forgot the connector.
Are you not going to be in today?
Oh, my God.
Oh, you got to pass on the...
Oh, here it is. Ah! Nope. So, cells, right? Cells know about the plasma membrane, know about the cell parts. Yes? Sugar.